Hello everyone, I am Dr. Amit. Welcome back to my YouTube channel at the rate Dr. Amit Official. If you are new here, please do subscribe for more simple and high-yield medical lectures. So in our previous video, we talked about electron transport chain, oxidative phosphorylations, and we talked about the 5 complexes, complex 1, 2, 3, 4 and 5. Okay, and how the electron is getting transferred and how ATP is getting produced. Okay, now in this part, we'll talk about uncoupling protein. Okay, and we'll revise all the complexes together. Okay, so this is one of the most important topic which is most frequently asked in most of the entrance exam. So let's understand this topic and let's start our part 2 of ETC and oxidative phosphorylation. So let's have a quick recap from the last video. Okay, so we all know that ETC and oxidative phosphorylation is a final pathway where NADH and FADH is producing ATP. Okay, now that NADH is giving electron to complex 1, FADH to complex 2, then the electron transfer to coenzyme Q, then complex 3, then cytochrome C, then complex 4, and at last complex 5 make ATP. Okay, so this is all we talked in detail in the last video. Okay, so now in this part we'll talk about uncoupling protein. So this uncoupling protein is very important concept in oxidative phosphorylation. Okay, so actually it helps in a specific alternative pathway. Okay, so normally what happen in ETC, that proton gradient is created and used by ATP synthase to produce ATP. Okay, but with uncoupling protein, okay, proton gradient, okay, is created, but it bypass the ATP synthase. Okay, and it helps in heat productions. Okay, heat productions in the body. Okay, how? By increasing inner mitochondrial membrane permeability, and it decreases the proton gradient and helps and then ATP synthesis stops, but the ETC continue. Okay, we'll see here. Okay, so we can see in this figure. Okay, actually what happened in uncoupling protein, that whatever, okay, this proton gradient, we all know that the proton gradient is being created and then again the proton moves to complex 5 and ATP generates. Okay, but in case of uncoupling protein, okay, they bypass this ATP synthase. Okay, they bypass this step. Okay, and by passing this step, complex 5, okay, it produces heat. Okay, so there are two uncouplers mainly, 2,4-DNP and brown fat in our body. Okay, so actually ATP is not producing, but energy is being released at heat. Actually, why this happen? Because sometimes our body need heat more than ATP. Okay, when to survive in cold or to maintain the body temperature. Okay, or we can also say that it is a protective mechanism in newborn to regenerate heat instead of ATP. Okay, so when our body needs more heat than ATP, so at that time what actually happened, this ETC continue, electron transport chain continue, but at this step, okay, at this step the electron will not go and make ATP. They will just bypass this step. Okay, bypass this step and produce more and more heat in the inner mitochondrial membrane. Okay, like in the body brown fats. Okay, so that our thermoregulation, or we can say that so that our body temperature will be maintained, or so that we can survive in the cold. It's also a protective mechanism in case of newborn child. So now this is the last from the oxidative phosphorylation. Okay, so we all know that one NAD is equal to 2.5 ATP and one FADH is equal to one NADH is equal to 2.5 ATP and one FADH is equal to 1.5 ATP. Okay, so NADH electrons from glycolysis enter mitochondria. We all know by to get, one is malate aspartate and one is glycerol 3-phosphate subtle. Fine. So actually this happen in aerobic conditions. So if the NADH enters through malate aspartate subtle, then it produces 32 ATP, which occur mainly in heart and liver. But if the NADH of glycolysis, okay, produced from glycolysis enters from glycerol 3-phosphate subtle, then it produces 30 ATP. Okay, actually it occur mostly in the muscles. Okay, but in case of anaerobic glycolysis, only 2 ATP is getting produced. Okay, now after calculation of ATP, one more important thing is that aspirin overdose. Okay, whenever there is aspirin overdose in any patient, then there will be uncoupling of oxidative phosphorylation. Okay, as we know that what's the main function of uncoupler, actually uncouplers work when the body needs heat more than ATP. So they actually omit this step. Okay, they bypass this step and produce more heat in this layer of our body, okay, inner mitochondrial membrane. So actually when aspirin overdose occur in a patient, there will be uncoupling of oxidative phosphorylation. So when there will be uncoupling of oxidative phosphorylation, then the ATP will not produce, and this will result in hyperthermia. So most of the time they ask in MCQ that why hyperthermia occur in aspirin overdose, because there will be uncoupling of oxidative phosphorylation. Okay, so at last I'll just quickly revise all these steps. Okay, so we all know that ETC and oxidative phosphorylation is the final pathway in the cellular respiration to produce ATP from NADH and FADH2. Then the electron get transfers, and they pumps protein, these complexes pump protein out, and they create a gradient and finally make ATP, right? So there are 5 complexes. One is complex 1, 2, 3, 4 and 5. Okay, NADH enter through complex 1, FADH enter through complex 2, then they transfer electron to COQ, that's coenzyme Q, then to complex 3, then cytochrome C, this NADH and this FADH directly to complex 4. Then they this proton again enter in the complex 5, and finally they make ATP, right? And this all occurring in the inner mitochondrial membrane. Fine. So this is all about ETC and oxidative phosphorylation. Okay, and I think you already know how we have calculated ATP, that why one NADH produce 2.5 and one FADH to 1.5, then how it equal to 32 and 30 ATP from different subtle. Okay, we all know this. Okay, so and always try to remember the what are the inhibitors of the different complexes. Okay, so it's very important in case from the MCQ point of view. Okay, so always try to remember those inhibitors. So for complex 1, okay, the main inhibitor is rotenone and amytal. For complex 2, the main inhibitor is malonate and carboxyl. For complex 3, the main inhibitor is antimycin. Okay, now for complex 4, the inhibitors are cyanide, carbon monoxide and azide, and for complex 5, the inhibitor is oligomycin. Okay, so this is all about ETC and oxidative phosphorylation. If you like this video, please do like and share with your fellow medicos, and please subscribe my YouTube channel @Dr. Amit Official. So this is all about ETC and oxidative phosphorylation. Thank you.